Here we perform total WBC count. Principle: a sample of blood is diluted with diluting fluid, which destroys RBCs and stain the nuclei of WBCs. The cells are counted in improved Newbar's chamber and the number in undiluted fluid is calculated. Starting with materials. For WBC count, we require microscope, WBC pipette. Here is the WBC pipette. Improved Newbar's chamber and cover slip. WBC diluting fluid, also known as Turk's fluid, and watch glass. Constituents of Turk's fluid. Turk's fluid contains glacial acetic acid, gentian violet, and distilled water. Procedure: with all antiseptic precautions, get a finger prick. Discard first 2 drops of blood and let a good size of drop to form. Here we are using collected sample of blood. Now, dip the tip of our WBC pipette in the edge of the drop and draw the blood to the mark 0.5. You can see here we have drawn the blood up to 0.5 mark. Suck the Turk's fluid to the mark 11. Mix the content of the bulb thoroughly for 3 to 4 minutes. Next step is move the Newbar's chamber to the microscope and focus the grid. This is the improved Newbar's chamber view under low power. You can see these 4 corner squares. They are WBC counting squares in the Newbar's chamber under low power. Without changing focus, now shift the Newbar's chamber and charge the Newbar's chamber. Place the cover slip. Discard first few drops of fluid from the WBC pipette and charge the chamber. The chamber should neither be overcharged nor undercharged. Allow the cell to settle for 2 to 3 minutes. Then carefully transfer the chamber to the microscope. Use fine adjustment again and try to identify WBCs. Counting the cells. Count the cells in 4 groups of 16 squares each. Total 64 squares. We are counting in low power, so this is the view of improved Newbar's chamber in low power. To avoid counting the same cells again, follow the rule of counting using L pattern counting. Count the WBCs present in the square and those on its left and lower line. You can see here inside the square and in the left and lower line, and ignore the WBCs present on right and upper line. Calculation for total WBC count. Volume of 1 square. As we can see, length of 1 square is 1 millimeter and breadth is 1 millimeter. Depth, that is 0.1 millimeter. So the volume of 1 square is 0.1 millimeter cube. Therefore, volume of 4 squares becomes 0.4 millimeter cube. In 0.4 millimeter cube volume, we counted the cell. Number of cells is n. N cells in 0.4 millimeter cube. In 1 millimeter cube, number would be n multiplied by 1 divided by 0.4. Dilution. Here we have taken blood up to 0.5 mark and diluted it up to 11 mark. As the fluid in the stent part, 1 part, does not take part in the dilution, our dilution is 0.5 raised to 11 minus 1 is equal to 0.5 raised to 10 or 1 raised to 20. You can see here dilution is 1 raised to 20. In 1 raised to 20 dilution, our count is n by 0.4. Therefore, undiluted blood, 1 raised to 1 dilution, the count is n upon 0.4 multiplied by 1 divided by 1 by 20. Therefore, the count is n multiplied by 20 divided by 0.4. Or you can say n multiplied by dilution factor divided by volume, and n multiplied by 50.
Here you can see in Newbar's chamber, when we are focusing it under low power, we can see 9 squares. From this 9 square, corner 4 squares, they are employed for counting WBCs, and they are known as WBC counting squares. These 4 squares are further divided into 16 small squares. So this is one of the 4 squares which is further divided into 16 squares. You can see here under low magnification, WBCs appear as round, shiny, you can say refractile, darkish dots with a halo around them. These dots represent the nuclei which have been stained by gentian violet. Do not confuse with dust particles which have varying size and shapes, often angular, and they are usually opaque with no halos around them. They are dark brown or yellowish in color. Counting the cells. Count the cells in 4 groups of 16 squares. This is one group of 16 square. This is second group of 16 square. Third and fourth one. So total we are counting cells in 64 squares. Total number of WBCs we have counted in all these 4 squares is equal to n, and when we multiply this n with multiplication factor 50, we'll get the WBC count.